uncovering the role of environmental chemicals in breast cancer etiology. I am Dr. Michael Mislak, medical advisor for the Massachusetts Breast Cancer Coalition, and I'd like to thank all of you for joining us today. Massachusetts Breast Cancer Coalition is dedicated to preventing environmental causes of breast cancer through community education, research advocacy, and changes to public policy. I am pleased to welcome today's speaker, Dr. Alexandra White. Dr. White is an environmental epidemiologist whose research focuses on identifying modifiable environmental exposures linked to cancer risk, particularly breast cancer. Her work examines how pollutants like polycyclic, aromatic hydrocarbons, and metals contribute to breast carcinogenesis, leveraging large-scale studies such as the Long Island Breast Cancer Study Project and Sister Study. She also investigates the role of epigenetics as a biomarker and potential mechanistic link between environmental exposures and cancer. Dr. White earned her M.S.P.H and Ph.D. in epidemiology from the University of North Carolina at Chapel Hill's Gillings School of Global Public Health and completed a postdoctoral fellowship at the NIEHS. She has been a tenure-track investigator with the NIEHS Epidemiology Branch since 2019. Dr. White, welcome. And one note, I guess, GoToMyPC is having some issues with their platform. So if you have any problems with logging on and back on, getting logged off, we're working behind the scenes to try and rectify that. So Dr. White, welcome.
Thank you so much. Thank you for the invitation to be here today and for the kind introduction. I'm really excited to share some of our ongoing work focusing on the breast cancer exposome. So my overarching research goal is to improve our understanding of how environmental exposures impact the development of cancer in women, particularly breast cancer. So I know this audience is probably very familiar, but I want to start with a brief background on breast cancer epidemiology. So breast cancer is the most common cancer diagnosed among women, both in the US and worldwide. It's estimated that over 300,000 new cases were diagnosed in the US in 2025. This translates to 1 in 8 women going on to develop breast cancer at some point in their lifetime. Importantly, in the last decade of available data, breast cancer incidence is on the rise. This is true for women who are diagnosed with young onset breast cancer, as well as those diagnosed at later ages. And this is really important because we've been doing a lot of work to try to identify risk factors for breast cancer, and it's really not yet translating to a reduction in incidence rates. And so this really highlights a key need to identify new modifiable risk factors for breast cancer. Of course, this is easier said than done, and is complicated by the fact that breast cancer is a very heterogeneous disease, with risk factors varying based on tumor subtypes. So we often define tumor subtypes based on the hormone receptors, estrogen and progesterone. Hormone receptor positive tumors are more commonly diagnosed, although less common hormone receptor negative tumors tend to have worse clinical outcomes, and they have very few established risk factors. And so one potentially important group of risk factors for breast cancer is environmental exposures. To estimate that we are exposed to over 350,000 chemicals that have been registered for production and use around the world. Yet, the International Agency for Research on Cancer, which is considered the gold standard of classifying a compound as a carcinogen, has really only classified about 1,000 agents based on their carcinogenicity. And a small percent of these chemicals have undergone toxicologic testing. So we really only understand the health effects of these chemicals really, for the tip of the iceberg here. So we're exposed to a vast amount of chemicals in our daily life, for which we know very little about the long-term health impacts, and even less about how these compounds may act together as mixtures to influence our health, including our risk of going on to develop breast cancer. And so this goal of understanding the totality of environmental exposures has been referred to as the exposome, and this is conceptualized to be a complement of the genome. And the goal here is to consider an individual's lifespan history of environmental exposures from in utero to death, and that this comprehensive understanding of the totality of environmental exposures is really important in understanding the development of disease. The exposome is a dynamic concept. It changes over our life course. And this can seem really daunting, the idea of trying to capture an individual's totality of environmental exposures. But I think that we can really implement this concept by using systematic and discovery-based approaches that help us generate new hypotheses rather than focusing on the same small number of known exposures. And the goal here is really to expand the amount of chemicals and the types of compounds that we can consider to really identify those new risk factors for breast cancer. So this figure shows the different domains of the exposome. So we have our external exposome. These are these exogenous factors that we experience in our daily life, such as exposure to outdoor air pollution and wildfires and the chemicals that are in our personal care products and other consumer products. We also have the internal exposome. And so the internal exposome can be broken into two different compartments. So the one compartment is these exogenous chemical biomarkers. So these are the chemicals that we can measure in our bodies that represent an internal dose of those exogenous exposures. We also can capture endogenous metabolites. And these metabolites can be a reflection of our body's response to those exogenous exposures. And the ability to really characterize the internal exposome and to capture environmental chemicals on a very large scale is really a recent technological advancement. So using targeted and untargeted high resolution mass spectrometry, we can capture this cumulative measure of the internal exposome and can identify hundreds of thousands of signals in a single biospecimen. And this can include both those external chemical biomarkers as well as our response to those stressors. And this technology gives us a really important opportunity to characterize exposure to previously unidentified chemicals and metabolites and identify new risk factors for breast cancer. So to do this, we use two different platforms, liquid and gas chromatography mass spec. So this figure shows the different chemical compounds that are captured across these two platforms. So gas chromatography, or GCMS, tends to capture more of those exogenous environmental chemicals, such as PCBs, PCDDFs, pyrethroids. LCMS has been more widely used for capturing metabolites, so endogenous metabolites. But LC also can capture some exogenous environmental chemicals, such as PFAS. So using these two platforms together, we can more widely capture a range of environmental chemicals to better characterize an individual's environmental exposome. So to do this work, I'm using data from the SISTER study, which is an ongoing prospective US-wide cohort. The cohort includes over 50,000 women who were enrolled between 2003 and 2009. Importantly, all women in the SISTER study have a sister who was previously diagnosed with breast cancer. And that puts our participants in the SISTER study at about a 2 times higher risk of going on to develop breast cancer themselves. At the time they enrolled in the study, women had a home visit with a trained examiner. They donated biospecimen and completed extensive questionnaires. This cohort is ongoing. We continue to follow these women. Our primary outcome of interest is breast cancer, but we also collect information on other cancer types as well as other chronic conditions. Women complete annual health updates as well as more detailed follow-up surveys every 2 to 3 years. For a subset of our participants, we had them come back between 2013 and 2015 and complete the baseline activities again. And this included about 1,200 women who had not yet been diagnosed with breast cancer. And so for these women, we also have biospecimen, as well as additional information about their home and their life at this time point. So for our analysis, we started with a pilot study. For the pilot, we selected 200 women for which we had these 2 blood samples. They were collected approximately 6 years apart. And these women had not yet been diagnosed with breast cancer. And the goal of the pilot was really to explore, to show feasibility of measuring these environmental chemicals in the blood and to look at stability of these measures over time. For the larger study, we conducted a nested case cohort sample. So for this, we selected about 2,000 women who went on to develop breast cancer over the follow-up period, as well as a comparison group of about 2,000 women that were randomly selected. As part of the study design, we oversampled for women who are diagnosed with hormone receptor negative breast cancer because it's less commonly diagnosed, and we wanted to improve our ability to look at risk factors for those less common tumors. We took these blood samples for both the pilot and the full study, and we sent them to our collaborators at Emory University, where they conducted targeted and non-targeted GC and LC high-resolution mass spec. So from these assays, they were able to identify over 360 unique chemicals with high confidence, meaning that we're very confident both in their identity and we can quantify the exposure levels to those chemicals. In terms of what was highly detected, over 159 chemicals were detected in at least 80% of our participants. So to give you an idea of the sister study baseline characteristics, so the women were on average about 55 years of age at the time they enrolled in the cohort. The cohort is 9% Black, 5% Hispanic, and 84% non-Hispanic white. And this is a slightly higher socioeconomic status cohort, so about half of our women have a bachelor's degree or higher, and a third had an annual household income greater than $100,000 a year. And this is similar to other volunteer-based study populations in the US. So our goal here is to use these exposomic technologies to characterize the internal blood exposome to give us additional to cast this wide net and give us additional information about how environmental chemicals are related to a woman's risk of going on to develop breast cancer. So coming back to our diagram here of the exposome, this is a large amount of data. So we have hundreds of thousands of different chemical signals and peaks in the data. And so we decided to focus our research questions and to start by really looking at those environmental chemicals that we thought were plausible breast carcinogens. And so to define, these breast carcinogens, we used a recent report that came out from the Silent Spring Institute. And what Silent Spring did is they used in vivo, in vitro, and in silico data from IARC and EPA databases to identify 900 potential mammary carcinogens. So these are compounds that can induce mammary tumors in rodents and chemicals that can act as endocrine disruptors. And so of those 900 chemicals, we were able to identify 200 of them in our sister study participant blood samples. We focused on those that were more highly detected. So we had 76 chemicals that were detected in at least 60% of our participants. So this figure shows the associations between those 76 potential breast carcinogens with overall breast cancer in the sister study. So this is what we call a volcano plot. And so the individual dots are point estimates for these single chemicals. So the hazard ratio is on the x-axis. So values that are farther to the right have stronger associations. And then we have the log of the p-value on the y. So the dots that are higher up are more statistically significant. So I'll walk you through some of these findings. So our most strong statistically significant association is for progesterone. And progesterone is not an exogenous chemical, it's an endogenous metabolite. And we did include it here in our analysis because it was part of the Silent Spring list. But this is very confirmatory because we know that circulating progesterone levels have been related to breast cancer risk in other study populations. I want to point out that our two strongest signals were for two pesticides, bromosil and carbofuranfenol. Bromosil is a broad spectrum herbicide that has been classified as a possible carcinogen. Carbofuranfenol is an insecticide, which is actually one of the highest acute toxicities to humans, and actually resulted in it being banned by the EPA around the time of our blood sample collection, although I think it still is currently in use in certain farming sectors. We also see positive associations for a phthalate here, M.E.H, as well as for nicotine, which is a marker of cigarette smoking. So when we repeat this analysis focusing on hormone receptor positive breast cancers, which make up the largest proportion of our cases, we see a very similar volcano plot with a lot of the same chemicals coming up, but we do see some additional hits. So we see a positive association for pyracrezidine, which is an industrial chemical that's used in dyes and is present in food and textiles. This is one of the known contaminants of red dye 40. And the National Toxicology Program has classified this as a likely carcinogen. Also, interestingly, we see a significant positive association for exposure to retinoate. This you may have heard is referred to as retinol. This is a derivative of vitamin A, and it's very commonly used in skincare products. So that was very interesting. And so we know that women are exposed to not just a single, environmental chemical at a time, but these exposures are correlated and people experience multiple exposures. So we also wanted to ask the question about the joint effects of being exposed to multiple suspected carcinogens. And so we used a method called quantile-G computation to ask the question of if you increase exposure to all of these suspected carcinogens, what is the relationship with breast cancer risk? And so what we see is a strong positive association for overall breast cancer, where increasing exposure to these suspected carcinogens is associated with a hazard ratio of 1.5, about a 50% higher incidence of breast cancer. We see a very similar association for ER-positive breast cancer, and our associations for ER-negative tumors was attenuated and not statistically significant, but still elevated. So this study is really important. Many of these chemicals have not been previously investigated, despite their biologic plausibility for being relevant for breast cancer. They haven't been looked at in epidemiologic studies to evaluate relationships with breast cancer. So in starting this project, we were really excited about focusing on these exogenous chemicals, and we started with these plausible breast carcinogens. But we're also really interested in PFAS exposure, and PFAS are per and polyfluoroalkyl substances. So you probably have heard of PFAS before. These have been referred to as the forever chemicals. And this is because they have the structure of PFAS has this very strong carbon-fluorine bond, which is very persistent and makes it really difficult to degrade both in the environment and in our bodies. This is a very large chemical class. It includes over 15,000 compounds, although we only routinely measure and study very few of them. We are exposed to PFAS in contaminated water, as well as in our diet. It's in our food packaging and in our consumer and personal care products. These compounds are truly ubiquitous, with almost all US adults having detectable levels of multiple PFAS compounds in their body. So I served on the IARC working group where we recently reviewed the evidence for PFOA and PFOS, which are two legacy PFAS compounds. And we classified PFOA as a group 1 carcinogen, which is the highest confidence in terms of carcinogenicity, and PFOS as possibly carcinogenic group 2B. However, these decisions were based largely on mechanistic evidence and animal studies, as well as some epidemiologic studies with other cancer types, whereas for breast, the evidence was considered insufficient. And around the same time, my research group led a systematic review and meta-analysis where we looked at the literature for PFAS and breast cancer. And we also really highlighted the need for more prospective studies, those that are collecting blood samples prior to diagnosis, and particularly those that are large enough to look at the differences by tumor subtypes. An important thing to keep in mind about PFAS is that we've actually known that PFAS are potentially harmful for quite some time. So the legacy PFAS, is, in particular PFOA and PFOS, which we reviewed with the IARC report, were actually phased out of production in the early 2000s, and they've been replaced with new and emerging PFAS compounds. And we can actually see the effects of this replacement in our data. So if you'll remember, I talked about our pilot phase of our study, where we collected data from 200 sister study participants who had paired blood samples 6 years apart. So we're looking at the same person 6 years later. And so we quantified PFAS exposure in both the baseline and the follow-up blood samples. And so we did this using a targeted assay, and we were able to identify 20 legacy PFAS compounds. And as we would expect, based on what we know about the phase out, we see lower concentrations in general of these PFAS compounds at that second time point. However, despite this trend of a declining exposure, we also still see that these are very highly correlated measures, which really speaks to the persistence of these compounds in our bodies. In our untargeted LC data, we were able to identify over 1,800 features that were annotated as PFAS compounds. So a really stunning amount of replacement PFAS compounds. And for many of these, we see what we would expect, which is higher concentrations at that second time point. But these compounds, they tend to be more short chain PFAS and they're possibly less persistent. So we see that these measures are less correlated and we see more variability over that 6-year time period compared to the legacy compounds. So our study was really ideally timed in terms of when our blood samples were collected to really see, I think, some of the effects of the legacy phase outs because we're able to detect both the decline as well as the rise of the replacement compounds. So it's really clear that we are being exposed to a wide amount of replacement PFAS compounds, but we know very little about the potential health effects of these compounds. So in our breast cancer study, we focused first on those targeted PFAS compounds. So these are largely legacy compounds. And we were able in our case cohort to detect 13 of these PFAS compounds. And so this figure shows a log increase in each of these individual PFAS in relation to overall breast cancer risk. And we don't see evidence of very strong associations for many of these PFAS. We see some suggestive higher risks associated with exposure to PFDA, which has actually been observed in a few other studies, as well as PFOSA. But overall, no statistically significant associations with overall breast cancer. But when we consider this relationship stratified by estrogen receptor status of the tumor, we see a very consistent story of stronger associations for ER-negative cancers. And this was particularly true for PFOA and PFOS. So you can see here that women who had higher quartiles of PFOA and PFOS had a statistically significant stronger risk for ER-negative breast cancer, and these are significant P4 trends and significant hazard ratios. So these findings are currently under review at a journal, and I think this is a particularly important study because it is both the largest study to look at PFAS and breast cancer, and it includes more PFAS than previously any studies had done. But most notably, we have the most number of ER-negative cases here, and this is really where our findings I think are most impactful because many previous studies have not had the ability to really look at this association by ER status and be powered to do so. So, you know, hopefully these results will be in publication soon. And so right now what we're really focusing on is better understanding the relationship between those emerging and replacement PFAS compounds and breast cancer risk. So we have identified over 2,000 compounds that have been annotated as PFAS in our breast cancer case, nested case cohort study. And we're exposed to these PFAS as mixtures as well. We see high correlation between certain PFAS compounds, which could indicate similar exposure sources or you know, co-exposure patterns. And so we use an approach called network analysis to help visualize the relationship between these individual PFAS compounds. So this figure shows the 10 different mixtures that we identified, 10 different networks of PFAS compounds. So each dot represents a single replacement novel PFAS compound or legacy compound in this, but it's predominantly the replacement PFAS compounds. And so you can see here, we identified 10 different mixtures of varying size. So mixture 1 is the largest with over 100 PFAS, and then the mixtures get smaller. And so we were interested in understanding how joint exposure to these replacement PFAS mixtures was related to breast cancer risk. So using the same approach that we did in our silent spring analysis, we used a method called quantile G computation to estimate how increasing all the PFAS jointly within each mixture was associated with breast cancer risk. So this figure, this first panel shows it for overall breast cancer, and the second panel by ER status of the tumor. And you can see that overall, these PFAS mixtures don't appear to be strongly related to breast cancer until we again stratify by ER status. So we see some evidence of stronger associations, again, for those ER-negative tumors, so consistent with what we saw for the legacy compounds. So we see elevated associations for mixture 8, mixture 2, and mixture 1, with a statistically significant association for mixture 2. And when we try to dig in, so there was about 50 PFAS compounds in mixture 2. So when we try to look in more detail about what makes up, you know, what PFAS are part of this mixture, there's really limited information on these compounds and very limited testing information. And so this is, you know, a really challenging area of study because these PFAS compounds, you know, there's so many of them and they're being phased out very quickly. But we certainly need to better characterize our total exposure to these replacement compounds and understand their potential health effects in addition to the legacy compounds. And this is the first non-targeted study of these replacement PFAS compounds in relation to cancer. Okay, so moving forward, so we've really focused so far on how we can use these measures to capture exogenous chemical biomarkers and how they're related to breast cancer risk. But we're also really interested in using these biomarkers to better understand how it reflects our external exposures, and we've particularly been focusing on exposure to wildfires. So wildfires, as I'm sure we're all familiar with, are increasing across the US and are increasing in their frequency, their duration, and their intensity. And wildfire smoke is projected to be one of the dominant sources of exposure to fine particulate matter in the coming years. And so we've been doing some work looking at how wildfire smoke exposure is related to breast cancer risk in the sister study cohort. And so to do this, we've used a model from Childs et al., which is a machine learning model that incorporates information on ground, satellite, and smoke plume data to estimate ground-based daily wildfire PM2.5 concentrations on a 10 by 10 kilometer grid. And so we linked this to sister study participant addresses starting in 2006 and following up to 2020. And because this is a really new area of research, it's not really clear what the best measurement is to better understand wildfire smoke in relation to chronic health outcomes. So we looked at a handful of different measures, including mean wildfire PM2.5, mean wildfire PM during peak weeks, as well as looking at smoke days and smoke waves, which are defined as being 2 or more consecutive days with wildfire smoke. And so these metrics get at different combinations of looking at frequency, duration, and intensity. And so we first look at how long-term wildfire PM2.5 was related to breast cancer risk in the sister study. And so what we see is some suggestive, positive associations for women who experience more wildfire PM2.5 in relation to breast cancer risk. And this is particularly notable for smoke waves. So women who experience more than one smoke wave compared to those who experienced less than one had about 7% higher risk of overall breast cancer. And this association seemed to be driven by those diagnosed with ductal carcinoma in situ or DCIS stage 0 disease, as well as those with ER-negative tumors. So this work is very timely, I think, given the public health impact of wildfire smoke exposure, and is also the first study to consider how wildfire smoke may be related to breast cancer risk. But getting back to our exposomics measures, so we use the same exposure data to look at blood chemical levels in our sister study participants. So this figure, again, we're back to that volcano plot where the individual dots represent individual chemicals. So this figure is showing mean wildfire smoke concentrations in the 3 months before blood draw in relation to blood chemical levels. And interestingly, what we see is that higher wildfire PM2.5 exposure is associated with higher levels of 8 chemicals at a raw p-value of 0.05. And what most notably to me, what we see is really a higher level of a number of different pesticides, including carbofuranphenol, toluene, as well as industrial chemicals, pentachlorophenol, PHPA, which is a PFAS compound. And I think this is, it's surprising, but then again, it also makes a lot of sense. Wildfires are not just burning, trees and organic matter, but they're burning agricultural areas, they're burning our built environment. And so these chemicals that exist in those environments are being volatilized into the smoke and maybe creating a more toxic air pollutant mixture. For example, pentachlorophenol is a wood preservative that's found in utility poles and railroad ties. And this has been classified as a potential carcinogen. We also see an association for carbofuranphenol, which it was one of the chemicals that we previously saw in our analysis to be related to breast cancer risk. So these findings are, I think, really interesting and one of the first characterizations of blood samples in women who have been exposed to wildfire smoke. So far today, I've really focused on those chemicals that we can identify with very high confidence in the data. But one of the really exciting things about this data is that we have a lot of other, chemical peaks that we may have less confidence in, but really have not been previously studied. So one of the things we're doing right now is looking at this non-targeted data. So we can look at how these mass spec peaks, or the kind of pattern of exposures in the data for the cases, are differ from those women who didn't go on to develop breast cancer. And then we can work with our lab colleagues to help identify those specific peaks that most strongly differ based on future breast cancer case status. So this is challenging because, certain chemicals may be difficult to confirm identities, but I think this is a really important aspect of this work, because I think it's really going to help us, identify potential new environmental chemicals that we may not even know we're being exposed to, but may be contributing to breast cancer risk. So I want to talk about a few things just to kind of put this data into context. So it's an important reminder that we're looking largely here at one blood sample at the time the women enrolled in the cohort. And so this is a strength of this study design is this is on average about 7.5 years before women developed breast cancer. But this is just one time point, which may not be the most relevant for breast cancer. We know that breast cancer, has many risk factors that are, in early life or in the reproductive window, and we're really not capturing that exposure. For some of these chemicals, like the legacy PFAS, which are really persistent, this one blood sample measure may be a very appropriate long-term marker. But for other chemicals, they may be more volatile or more variable in our bodies. And so we want to, put those findings into context for each specific compound. Here we use a large nested case cohort design, which is, the sample size of the study is a really important strength. It gives us the ability to explore differences by estrogen receptor status of the tumor, which was really important in a lot of our different analyses. And we're also able, because of the design, because we have this random comparison group, we're able to explore other very interesting research questions like the wildfire smoke and blood chemical levels. And so I think that makes this study really have a lot of other applications that we're excited to collaborate on and explore in the future as well. Some of the challenges of this large study design was that it took a very long time, particularly in data processing and quantifying exposure. The labs had never worked with a sample size this big, and so we definitely ran into unexpected challenges. One of the strengths of this is the first study of its kind to really implement this concept of the internal exposome for breast cancer. But these are the first findings, and so there's a real need to be able to replicate these findings in other populations. And so I know that people are interested in this research area and are putting funding proposals in. So I hope that in the future we'll be able to collaborate with others to replicate our findings and to strengthen the public health impact of this work. So one of the future directions is really integrating across multiple omes. So in this data, we have the exposome, we also have metabolomics data, we collect information on, we have epigenetics and genetics data. And so, I think where we're really going in the long term is being able to integrate across these different levels of data to better understand, why certain environmental exposures are related to breast cancer risk in some women, but not in others. And maybe that'll be an influence of genetics and epigenetics, but I think that will help us better understand the underlying biologic pathways. So I want to end today just talking a little bit about the significance and impact of this work. This is the largest targeted and non-targeted application of the exposome concept with respect to cancer to date. Our preliminary evidence highlights a role for pesticides, industrial chemicals, as well as personal care product-related chemicals in breast cancer development. And I just want to say that, this work is very important, but it's also very confirmatory of what we've been able to see in other, epidemiologic, studies where we've looked at, farm workers or, occupational exposure and industrial chemicals and self-reported personal care products. So it really, fits with the literature that suggests that these, are important sources of potentially harmful chemicals. But one of the benefits here is really, trying to identify those specific pesticides and industrial chemicals and components of personal care products that may be most strongly contributing to breast cancer risk. And as I mentioned, our future work will really delve more deeply into the non-targeted features to identify new chemical risk factors for cancer, as well as incorporate our metabolomics data to help provide evidence on potential mechanisms and metabolomic pathways specifically that may underlie these associations. And the real goal here is to highlight targets to really facilitate interventions that can decrease exposure to harmful chemicals, with the goal of really reducing the incidence of cancer in women. With that, I want to say that the sister study has a really great website with information about the study and all the data we have. So there's a lot of different collaboration opportunities. At the NIEHS, we have a great summer internship program, as well as options for pre and postdoc fellowships. I have my email here. Please feel free to reach out. And I just want to end with thanking my collaborators, particularly my postdoc students, present and past, Jenny Ish, Katie Paul, and CJ Chang, who are really doing the hard work of analyzing this big data, as well as our extramural collaborators, Doug Walker and Dean Jones at Emory, who ran the lab essays, and most importantly, the sister study participants, without whom none of this work would be possible. And with that, I'm happy to take any questions.
Great, thank you very much, Dr. White.
Yeah.
That was a terrific presentation.
Great.
I'd like to invite the audience to submit questions in the chat box, and I will read them off. We already have one question here.
Okay.
Could you please speak more about progesterone? Had the women been taking it, or was the body producing it? And what about estrogen levels? The listener thought that was a higher risk than progesterone, which she believed was more protective.
Yeah, so we haven't, so these women are pre and postmenopausal in the sample. So it's definitely possible that they could be taking some, hormone replacement therapy. And, we didn't capture estrogen in the data. So we weren't able to really disentangle that, but I think it's an important question. It's something that we need to look at in a little bit more detail. I think that I'm trying to remember, we did look at like self-reported use in relation to progesterone, and it was higher. So yes, I think that's very important question. And we've been focusing more on the exogenous chemicals in the data. But certainly I think there's room to use this as well to explore how, medication and, use of more probably hormone replacement therapy here than oral contraceptives, but could be contributing to these circulating levels as well.
That's great, terrific. As a pathologist, I certainly diagnose too much breast cancer, and it seems like more and more these days, and more younger patients with more aggressive cancers than I had in the past. And I was particularly interested in your Venn diagram that showed the exposome and sort of the biological circle intersecting with the external and the personal. And just from personal experience, I kind of function here in the biological circle, and just looking at under the microscope 2 different cancers that may look the same, and we try to use sequencing and tests like Oncotype to try and predict behavior and prognosis of tumors. And there's still so much to learn on the biological part, and that's still a small sliver of the entire exposome. So there's quite a bit still to learn. It's pretty exciting though.
Yeah, it is really exciting. I think you're right. Like there's so much to learn. And one of the things I think we can, I get asked a lot, we're using measures in blood here, but it would be great to be able to see if we can measure, these chemicals in the breast tissue. And, thinking more about, the microenvironment and how that could be contributing, to tumor development is really important.
Right. A few other questions have come in. How do these methods allow us to detect previously unidentified chemicals?
Yeah, so what I focused on here, as we just got this data, in the last year, so we've been, really focusing on those compounds that we are confident in. But one of the strengths of this untargeted methods is that, you're searching the, mass spec for not a specific peak, but for, any peak, and then you link the peak for, to databases that have been previously used to identify chemicals. And so it's a way to look at these blood samples without coming in with a strong, we kind of do both, right? We come in with a targeted list of compounds that we are able to measure, and we compare it to a standard, but we're also able to measure chemicals that we don't have a standard for, but by linking it with other databases, and then we can go back and confirm them, the existence of these compounds. It's a way to to capture more broadly these different environmental chemicals. Oh, I'm sorry, I can't hear you.
Sorry. And how about looking at the effect of timing on exposure of these chemicals in the patient's lifespan?
Yeah, so I mentioned a little bit, we're focusing on kind of this arbitrary time window of when someone enrolled in the cohort, but we know from other studies that there's periods of a woman's life, such as the reproductive period or puberty, that might be more relevant, particularly with an interest at early onset cancers. And so I think it's important, it would be an ideal future study to really kind of target our blood sample collections at those time windows, or get at earlier windows of exposure, because I think those are going to be when women are experiencing a lot of, for example, personal care product related exposures. And so I think that that's a strong future goal for our research, is to have a different study population that we can do that in.
Great, thank you. And we have one other question that's come in. In the wildfire smoke example, what do we know about the specific fires and what was burning?
Yeah, so we've thought a lot about that, because we're, so sister study participants are all across the U.S. And even though we know women in the Western U.S. are experiencing these wildfires, in general with more intensity and they're in closer proximity, we know that, the smoke travels. I'm in North Carolina, and, we don't have a lot of wildfires here, but we do when there are major, the Canadian wildfires 2 years ago, we experienced the smoke here as well. So, this is, kind of a cross section of the US, and we're not able to really map back to specific fires, but I think that could be a really important, consideration for future studies of wildfires, because the type of contaminants that are in the smoke are really going to vary, whether it's an urban fire or in more agricultural areas. And so I think by better characterizing individual fires, we'll be able to better understand the potential, health impacts of the smoke.
Wonderful, thank you. I think that's all the questions that came in. Certainly a lot of interest and discussion online, and I think it's a great topic, and certainly a lot more to find out. I'll be closely following along with your research, so thank you very much. On behalf of myself, the MBCC Board of Directors, and Cheryl Osmo, MBCC Executive Director, I want to thank Dr. White for this very informative discussion today. And I want to thank all of our many listeners for joining us. For those interested, the recording of this webinar will be made available later today on the MBCC website at mbcc.org. And if you'd like to further support our mission of preventing environmental causes of breast cancer, please consider making a donation by visiting mbcc.org/donate. I appreciate your consideration. Have a great afternoon, everybody. Thank you.
Thank you.